to start recording. Very, very happy to welcome this autumn speaker, Professor Natasha de Vere. Natasha is currently the curator of botany and professor at the Natural History Museum of Denmark in the University of Copenhagen. Natasha has been there about 3 years or so. And before that, she was based in the UK, in Wales, at the National Botanic Gardens in Wales, where she was there from about 2007, 2008.
Yep.
Yep. And in the National Botanic Garden of Wales, she was the head of science, and also, I think a couple of years later, became a senior lecturer at the University of Aberystwyth. Mm-hmm. Yeah, in Wales as well. Natasha did her first degree, I think, in Oxford, and then I learned a master's at the Royal Botanic Gardens in Edinburgh. Yeah, Laura. And she, I think, throughout her career, Natasha has been a pioneer in the use of genetic and molecular tools to study plants and plant diversity. And it is, of course, my interpretation. I'll let her to correct me later. But one of the things that I think Natasha made a big contribution on was in 2008 or so, she developed this large-scale project to barcode the flora of Wales, every single vascular plant, and then it was expanded to the whole British flora. And I think that project took several years to complete, but what was completed and became one of these flagship projects where molecular tools are used to understand better the flora of an entire country. Now, Natasha has a very diverse interest. She's interested in plant-pollinator interactions and how to use ecological and genetic tools to study these interactions. She's interested in how climate change is affecting plants and their interaction with pollinators. She's also interested on aspects of how anthropogenic factors affect the diversity of plants. I think initially in the UK, but now in Denmark and Greenland. I think she's going to tell us a little bit about her recent work there. But the unifying theme is this approach, this first revolutionary and now still path-leading approach to study plant diversity with a combination of molecular tools. However, Natasha is also interested in diverse approaches to botany and science, and I know she cares about how people perceive plants in her role in the botanic gardens in both the UK and Denmark. But I also heard that you have some interesting projects in the intersection between arts and science, and I'm not sure what they are, but maybe over dinner we can ask you more about it. I want to thank you very much for coming to visit us and deliver this talk. Also because you have hung out with us the whole day and interacting with other society members and members of the department. I very much enjoyed your visit, and I'm looking forward to your talk. Thank you.
Thank you. Thank you so much. Right, so thank you so much for inviting me along. I have had a lovely day in Uppsala with the sunshine as well and blue skies. And it has been lovely to meet so many of you, and hopefully to meet a few more of you this evening as well. So thank you for the introduction. So as Maria has mentioned, I've been at the Natural History Museum of Denmark just for 3 years now. And then prior to that, I was head of science at the National Botanic Garden of Wales. And I'm going to talk to you about some of the research that we've been doing over those years. And it's going to very much kind of divide into some of the work we did in the Botanic Garden in Wales, which has results, and then an introduction to some of the work that we're starting with my research group at the Natural History Museum of Denmark. And that's more the kind of direction that we're going to take. And most of that work is taking part in Greenland that I'm going to tell you about. But the theme is very much plants and pollinators. So my background is botany, and I'm curator of botany in the museum. But my research interest, particularly in kind of latter years, has been on plant-animal interactions, particularly with insects. So it was working perfectly and is now, okay. So first of all, why plants and pollinators? I mean, we know that pollinators are obviously both important, but they're also threatened. And they actually face a perfect storm of interacting factors that cause problems. Pests and diseases, loss of habitat, climate change, insecticides, and agricultural intensification have caused a decline in pollinators and ill health in honeybees. And one of the key factors that we know is a problem is a loss of flower-rich habitat for foraging. And so that means if we can find out more about that, if we can find out more about what plants are important, maybe we can do something about providing those in the environment. So the type of work that I'm interested in doing is, well, first of all, what plants are used by different pollinator groups. We've done lots of work on bees and hoverflies. We're doing some more work now on different pollinator groups in Greenland. And then how do plant pollinator networks change over time, whether that's over a season or whether that's over historical time? And what does that mean for how resilient those interactions and networks are and will be in the future? And I'm interested in how rapid environmental change impacts the interactions as well. So, for example, climate change, some of the work that I'm going to tell you about that we do in Greenland, but also within gardens and urban areas where we create quite artificial habitats and then pollinators come in and use them. But the background to all of this is how can we use this information to help to conserve plants and pollinators. But before we can do any of that, we just need to be able to find out what plants pollinators use. And of course, we can use observation. So we can look at a bee, we can follow it through the landscape, we can see what flowers it lands upon. But actually, that just gives us part of the story. We can't watch pollinators all of the time. We can't see what they do if they go up into the trees or beyond through hedgerows, or if we're working in a difficult environment to track them. So an alternative is to look at the pollen on their bodies and identify that as a means of understanding foraging. And you can identify that pollen either using microscopy, but we identify it using DNA. So first of all, we retrieve the pollen from the bodies of insects, for example, like here, or from pollen loads on bees, or within honey samples. And we extract the pollen from that and identify it. And the technique we use is called DNA metabarcoding. And using this, we can increase the temporal and the spatial coverage compared to observation. So we can do much bigger studies in terms of foraging. We can also increase the taxonomic resolution of the plants that we see. And we can use it to track individuals. So we can take an individual and see a complete record of the foraging trip that it has had on that day. So the examples that I'm going to give you of some of the work that we do all uses more or less the same methodology when it becomes to the DNA work. So I wanted to just give you a little bit of background on what that DNA work looks like and what the approach is. So first of all, we sample our pollen either from the body of an insect or from a honey sample. We extract the DNA from that sample, and then we increase two small regions of DNA called barcode markers. And we use two short regions of DNA. So we amplify those, and then we sequence that using next generation sequencing. And what you end up end of that process is a mixed bag of pollen. It's often a mixture of different pollen from different plants, and DNA from all of those in just a big kind of pile, but we don't know what those plants are at this point in time. So the ability to use this technique first requires a reference library that we can compare that DNA to. And the ability to make a good reference library of all of the different plants and their DNA barcodes is what makes the difference in terms of making this technique work really well. So a lot of the examples that I'm going to show you for the first half of the talk all come from the work that I did in the National Botanic Garden of Wales. And we used the Botanic Garden as a study system over many years. But the first thing we needed to do was make our DNA barcode reference library. So first of all, we made a DNA barcode reference library for all of the plants of Wales, and then we extended that to the whole of the UK. And we were the first nation in the world to actually have a complete library of all of our flowering plants. And we use that for many different applications. We've done projects on looking pollen in the atmosphere and relating that to hay fever sufferers. We've done projects which are identifying plants from soil samples. My particular research focus is on plant pollinators, which we'll be talking more about here. But you can also do things like, once you've got DNA for all of the plants of the UK, you can make a phylogeny of all of the plants. So this is a complete phylogeny of the UK flora that we made using the barcodes. And in this particular example here, we were looking at levels of hybridization in the UK flora and looking at what were the indicators for hybridization in the UK flora. So by creating these libraries, we can use it for many, many different applications. But our application is on plant pollinator interactions. And we started off doing a lot of work on honeybees. And this was partly because they were very easy to study. Once you have your hive, you know where it is, so you know where the bees are coming from. You can collect honey samples quite easily. And that provides a kind of overview of all of the different plants that a honeybee has been foraging on. You can collect those samples from the top of a comb, and you can find samples which have been freshly collected. But in our first studies, we looked a little bit more broadly, and we looked at historical changes in honeybee foraging across the whole of the UK. And key to this was we found a data set that was collected by somebody called A.S.C. Deans in 1952. And he did a massive survey of 885 honey samples that he collected from beekeepers all over the UK. And he identified that pollen from the honey samples, this time using microscopy. So we repeated that survey in 2017. We did a call out to beekeepers who sent us honey samples, and we identified it with DNA barcoding, and then spent quite a lot of time kind of cross-calibrating the results so we could compare between the different methods. And so we could compare, okay, what were honeybees doing in 1952 compared to 2017? And the chart on the right-hand side here, it doesn't matter that you kind of can't see the names on the left-hand side here, but it's basically a list of all the different plants that we found in the honey samples. And 1952 results are here, and the 2017 results are here. And the width of a bar is the frequency of honey samples that that plant was found in. And then the colour is a measure of abundance of how much of that particular plant was in the honey samples, with red being lots of it and going down to green if there was a lot less of it. And what you can see is that actually overall, there's quite a bit of similarity between the plants used by honeybees in 1952 compared to 2017. And the same 4 plants appear in over 50% of the samples across all of the years from 1950s and 2017. So Rubus fruticosus is important, Trifolium repens, Brassica napus, and a group which includes Crataegus and Malus, so hawthorn and apple, and the garden plant Cotoneaster. So those plants are important no matter what. But what we saw was actually a significant difference in the proportions of those plants used between the 1950s and more or less the present day. And those proportional changes in the plants that we saw reflect changes in landscape use within the UK, and also it would be the same across most of Northern Europe. So in the 1950s, Trifolium repens, which is there, was by far the most important honeybee plant. So if you give them a choice, white clover is really important for them. But by 2017, that had decreased significantly. It's still important, but it's been taken over in the top spot by Rubus fruticosus. And this makes sense. Within the UK, along with many places, from the 1950s and onwards, we then see a decline in pastures and meadows rich in wildflowers. So as we have an increase in artificial fertilizers and herbicides, an increase in grass lays, which are just monocultures of grass, which get cut regularly for silage, we see a reduction in the amount of white clover within the landscape. And the honeybees are balancing, we think, where they're using less white clover, they've replaced that with Rubus fruticosus. We also see a significant increase in Brassica napus, and this is the crop oilseed rape. And as a crop, it only really started to be grown from the 1970s or so and became really abundant from like the 1980s. And the honeybees have tracked that increase and use a lot more of it now that it's become available to them on a mass scale. This can obviously be problematic because it can also be a crop that's treated with neonic insecticides. And then we also saw an increase in an invasive species coming into the landscape. So Impatiens glandulifera, or Himalayan balsam, it's called in the UK, was first grown as a garden plant in Victorian times. And it's a big, tall plant, very impressive looking. So people loved it in their gardens, but it soon escaped. And it started to grow along watercourses, slowly at first, and now as actually quite an important invasive species. So first of all, it can outcompete other plants within particularly riverine systems. And when it dies, it's an annual, it can clog up watercourses. But it is also a really important pollinator plant towards the end of the season. It produces a lot of nectar and pollen, and many pollinators use it. And the honeybees have shown a significant increase in the use of it from 1952 to 2017. So with this technique, it's been a really nice way of basically looking at some of the landscape changes from the point of view of a honeybee within a managed colony. So we carried on looking at honeybees, and this time we wanted to look in more detail on the level of individual apiaries within one study site. So on this map here, this is our study site, which was in the National Botanic Garden of Wales, where I used to work. And the map on the top left there, that's actually the botanical garden. And next to the botanical garden was a national nature reserve, which is managed as an organic farm. And we mapped all of the plants that were found within these different habitats. This is a big, big site. The green is meadows. This is mostly species-rich meadows. The yellow is horticultural garden areas. And then brown is woodland and dark brown is hedgerows. And then we had two apiaries, one within the nature reserve and one within the botanical garden, but they didn't show actually any differences. So we've put all the data together. And over 2 years, we took 6 honeybee colonies and we looked at all of the different plants they were foraging on each month by DNA metabarcoding honey samples from the hives. And these charts show for 2016 and 2017, all of the different plants used from April through to September. And they actually use a fairly small number of plants. This black line here are plants that they use below 10%, so small amounts. So they use lots of plants, but often a lot of those just tiny amounts. And they have their real preferred plants that they use in abundance. And interestingly, it matches up with what we saw on a UK level. So by far the most important plant is this red one here, which is Rubus fruticosus, which matched the UK data. And then in purple here is Trifolium repens. So we can see that the pattern of plants used through the season for some honeybee colonies. We then took it another level again downwards, and we looked at an individual colony basis. So this is now data for 2019, but within the same study system of the botanical garden. And this time, this is April, May, June, July and August. And on the left-hand side, H1 to H6 are 6 different honeybee colonies. And all of the plants used for that month are on the right-hand side. And the width of the bar is the proportion of DNA within that sample. So it's a measure of relative abundance of the DNA. And what we can see is in April and May, the honeybees concentrate on spring flowering trees and shrubs. So they use a lot of Acer, they use a lot of Prunus and Salix early on in the season. And this makes perfect sense to what we would observe honeybees doing. But then something kind of a bit crazy happens in June, because the spring flowering trees and shrubs have gone over and other preferred resources of the honeybees aren't available yet. So each colony does something a bit different, and they use lots and lots of plants in small amounts. And so this actually reflects a period of resource limitation. And is there any beekeepers here? Okay, so because if I do a talk like this with beekeepers are like, well, yeah, of course, because you have the June gap. So it is well known by beekeepers that you have a gap in June. We've also done studies with bumblebees in farmland. Again, June is a period where there is a gap in resources. And we can track that by seeing how the networks change. So we have this period where they haven't got a single preferred thing, they're using lots of bits and pieces. We would also predict that it would be this point where you might have more competition between honeybees and other pollinators. Then by July, you can see something comes in flower that they love, Rubus fruticosus. So as soon as that starts to flower, they all migrate onto that, with a little bit of the purple, which is Trifolium repens. So honeybees are nice to study. They're very consistent in their choices, and you can look at these different spatial scales and see these patterns quite clearly. So then we thought, okay, let's move to wild pollinators. And this is still within the same study system. So this is the Botanic Garden and National Nature Reserve. But this time we wanted to see what bumblebees, solitary bees, honeybees and hoverflies were all doing within this landscape. So instead of sampling from hives, this time we had transects throughout the different areas, and each month we collected pollinators from these transects and looked at the pollen they were carrying on their bodies and identified that with DNA metabarcoding. And we wanted to see how the different groups of pollinators differed, but also how it changed over the seasons again. So this plot here each one of these dots is an insect, and it's the botanical composition of all of the plants on their body, which is plotted. And the plots on the green here from spring, and then these ones here are summer, and then these ones here are autumn. And it basically just shows a progression of the botanical composition of plants, which the pollinators were using. This is all of the pollinators together in one big group. And if we look at what those plants are, we can make a giant network, which has got all of the pollinators that we found on the left and all of the plants they used on the right. And this is data for 2 years for all the seasons, all put together. So this is a kind of snapshot of our nature reserve and our botanic garden for 2 years. So we've got honeybees on this bar here. We've got lots of bumblebees, lots of different hoverflies, and amongst here we also have a few solitary bees, but not so many. And the width of the bar is the number of those insects that we caught. And then on the right-hand side are those plants again, and the width of these bars is the proportion of the DNA. So across all the pollinators, there's definitely a few key groups which are important. So the umbellifers, so this is the Apiaceae with these nice kind of screen-like flowers for landing on, is very popular. Thistles are very popular. Buttercups, Rubus fruticosus, again, is very important. And in spring, dandelions, Taraxacum officinale, are important. And we can then look at the difference between the different groups. So were there differences between the hoverflies and the bees? So this graph here has got the hoverflies here and then all of the bees over here. And whereas there's some plants in common between the groups, there's also significant differences. So in green here for the hoverflies, this is the umbellifers in the Apiaceae, which are preferred by hoverflies, whereas the hoverflies like thistles, these ones here, the bees use a lot more thistles. And this makes perfect sense if you think about the structure of their mouth parts. We can also look inside the different groups of bees. So this is bumblebees and honeybees and solitary bees. Overall, as you would expect, you can see that bumblebees and honeybees are more similar with their plants, as solitary bees are doing all kinds of other things. Using the same data, we divided it into different functional groups. So for bumblebees, this is bumblebees with short tongues compared to those with long tongues. Again, different compositions. And this is honeybees here. So not surprisingly, short-tongued bumblebees are most similar to honeybees in the plants that they forage in. Hoverflies are here. So solitary bees, we have quite small sample sizes, but you can see that they're really variable. And we're really interested in solitary bees. There are so many different species, and we know so little about the plants that they use. So we're doing more work on solitary bees to find out more about them. But the results for hoverflies here are really interesting because these groups here are the larval's food source. So if the hoverfly larvae was carnivorous, detritivorous, or herbivorous, and you can see a totally different pattern of adult foraging compared to what the larvae used as their food source. The herbivore here, which is mostly used in buttercups, is known, it's mostly one species which is a specialist on buttercups. So we have a tool which we're just really beginning to delve into, which can give us a lot more insight into different pollinators and how those different groups are important and how those different plants are important. And for us, this particular sort of project, which was a number of PhD students over quite a few years, one of the main reasons of doing this within the botanical garden was to look at how can we improve gardens for pollinators. And so all the way through, whilst we were doing the research, we did lots of public engagement and advice to the public on pollinator-friendly gardening. We also had another PhD student, Lucy, whose PhD worked on designing seed mixes, annual seed mixes. So we tested commercial mixes that you can buy now, and then we made our own mix and we tested that to see if we could custom make one that was better for pollinators of different groups throughout the season. And we're just writing that up at the moment. And so we could say a few recommendations from the research in terms of what we could do in gardens for pollinators. So first of all, bees and hoverflies use some different flowering plants. Ensure that plant choice includes those preferences. Different plants are used through the year. So the beginning and the end of the season are really critical that we provide things in our gardens at that point. And the bees and hoverflies use both native and also non-native species. So giving space to the self-sown plants, the weeds in our gardens are really valuable. But garden plants are also important, particularly towards the end of the season, and it can extend the season for pollinators. And Rubus fruticosus came up as consistently a really important plant, so we should provide that within our landscapes. So one of the final outcomes of the project, we have got lots of scientific papers that we're writing, we're finishing up those. And then the idea is that I'll write a book, which is a kind of popular science book on plants for pollinators in gardens, which I'll get there eventually with producing. And then we did another piece of work, which was, again, how do we use this information? Because a big part of the work in the Botanic Garden, which I also want to continue at the Natural History Museum in Denmark, was not just to do this scientific research, but to use it very directly in ways which were useful. So obviously we could provide advice to gardeners, which we did lots of, but we also developed a Saving Pollinators Assurance Scheme. Because if you go to garden centres, sort of anywhere in Scandinavia or the UK, you'll find sometimes labels on plants that say good for bees or good for pollinators. And some research has been done by Dave Golson's group, actually, in Sussex. And they tested, okay, how well do those labels work? Also some work's done by Francis Ratniks. And they studied, okay, how well do those labels work? Or are they really better for pollinators? And they found that the answer was no, not necessarily. The lists and the labels are often based on anecdotal information. They then did some further work in Sussex where they tested some of these pollinator-friendly plants for levels of insecticides and found that some of them had levels of insecticides high enough to actually potentially harm pollinators. So we have a situation where, you know, the public is wanting to do their best for pollinators, but they're really being missold. So we got together with nurseries throughout Wales, and we got 29 during a pilot stage of this project altogether, who were already interested in growing pollinator-friendly plants in a sustainable way. And so we gave them our information on what plants were important. So they were often growing these plants already, or they could tweak their plant lists they were growing. But then we also had additional requirements. So first of all, the plants had to have been grown without synthetic insecticides. And they also had to have been grown without peat compost. So we weren't destroying a habitat to create our gardens. And if those were met, so if they were grown without peat and insecticide, and they were plants on our Saving Pollinators list, they could put the logo on their plants. And we looked at how well that worked. And in 21 and 22, obviously this is during COVID time, so lots of people were really interested in their gardens. This may also have helped. But on average, each nursery sold over 12,000 plants. And they stated that 83% increased their plant sales and 96% used that as a way of raising awareness. And these are not big companies. These are all mostly small local nurseries working within Wales. So like I say, this project is ongoing. We also want to start up some more work looking at the importance of gardens and urban areas for pollinators now that I'm in Denmark. But I also changed focus a little bit. So just in the sort of last quarter maybe of the talk, I just wanted to give you some examples of some of the projects which we're starting now with my research group in Denmark. And we were still interested in looking at plant pollinator networks, but we then wanted to look at the impact of climate change a little bit more. So we decided to start some work in Greenland. The Arctic as a whole is warming 4 times faster than the global average. The rate of change is so fast. And Greenland exemplifies that. We're probably all aware that eventually the Greenland ice sheet will go. If you look at levels of greening around the coast of Greenland, you can see the changes like incredibly rapidly. So we wanted to know, well, what impact does climate change have on plant pollinator diversity, but also plant diversity over time? And then we want to see how will those processes change and what can we potentially recommend to do about that. And a key point of starting this research and focusing on Greenland is because within Denmark, part of my role as curator of botany is I'm curator of the vascular plants, both within herbarium. So we're the keepers of Herbarium C. So we have around 2.3 million herbarium specimens that I try and look after. And in particular, we have an amazing collection of herbarium specimens from Greenland. And these have been collected from 1739 to the present day. And these collections have just been digitized. So we've just made these available for the first time, over 170,000 plant specimens. So what we wanted to do was be able to use this resource of this historical data combined with fieldwork that we could do in Greenland today. So if you think about the kind of example that I'd given in the UK, so the first thing was to make a reference library for DNA barcoding so that we can do our work there. But we actually had to take a step further back in Greenland because we found that all of the species lists and floras that we used all had different numbers of plant species for Greenland. The actual main flora by Buka is quite old now. And there's various lists with slightly different summaries of plants of Greenland. So our first thing was to make an up-to-date species list for Greenland based on the fact that we now had over 170,000 specimens that we could start to give the current accepted names to and use that to see, okay, what plants do we have in Greenland now? And my poor PhD student, Brandon, who thought he was going to be working on plant pollinator interactions, and he is, his first job then became to be a kind of like crash course in plant taxonomy, because he basically had to take all of these digitized records and sort out the accepted names for those, and then reclassify all of these records. And he's just about nearly done that. He's written an amazing R script that does all of this work and incorporates all of the information we can find for Greenland in order to make a species list from our digitized data, and also that available from other institutions as well. And so we'll send that for publication quite soon. So that was stage one. Once we've done that, we can then make a DNA barcode reference library for Greenland. And a bit like the UK, this is a really nice study system. There's around 700 plant species. And if we can understand this whole system of Greenland as an island, there's a lot of questions that we can ask about climate change and environmental change in the future. As well as making DNA barcodes, we will also make a genome reference library for the plants of Greenland. So we'll cover more of the genome of the plants. And then in terms of plant pollinators, these are some of the questions which we're just starting to answer. So we don't have results for these yet. We're just beginning to work on this, or really just starting to get funding in for some of these areas. So Brandon, the PhD student I mentioned, he's focusing on the effect of habitat and latitude on plant pollinator networks. We need to know just a little bit more about the kind of overall study system. So he's collecting pollinators from throughout Greenland and then starting to DNA metabarcode those to find out what plants are used. It's a bit difficult working in Greenland compared to the Botanic Garden in Wales. It's all a bit more expensive for first. There are no roads. And so this is like half of Greenland. Greenland is huge. It's a massive, massive island. And you can only travel around these places by plane or helicopter or military boat and things like this. So it's quite difficult to access these sites. We did our first field season in 2023, and we focused on something called Disco Island or Kirketassuaq, which is here because Copenhagen University has a field station there. And in 2023, we went there in June for our very, very expensive field season. And there was still snow, like this much snow on the ground in June. So we can't do anything. Like, you know, you bought your flights, you know, you're quite fixed. So we actually went to this place here, Kisiengarit. It's not very far away. It's like 3 hours on a boat. But the snow had completely melted, it pollinators everywhere. Then in 2024, Brandon went back with another PhD student. He visited a lot more field sites, but he also went back to these sites here. He went to Kirketassuaq, and it had nearly finished flowering. So this is exactly the same time of year. And this is what Arctic researchers are seeing. We're not seeing this kind of wholesale change in one direction or another. We're just seeing these extreme kind of stochastic weather events. So it's very, very difficult to study, and it's very scary in terms of like how extreme the weather is getting and how variable it's getting. But the other nice thing about this research is Greenlandic researchers, people doing this research, they tend to work together really well because it's also expensive and you have to fly to these places. So we would collect data for lots of other projects, and other researchers going to other sites would collect data for us. So a kind of combined effort enabled us over these years to visit all of these different sites and collect 1,772 pollinators. This is by far the biggest study that we will have of pollinators across the whole of Greenland. One of our main collaborators, who is based in Sweden as well, Thomas Roslin, he has done a lot of work on Zackenberg, and he's one of our collaborators on this. He's done lots of work understanding plant pollinator interactions in this site. We're now broadening out across a much broader area. So that's a kind of watch this space. So Brandon will be analysing that data over the winter, and we'll start to have our first plant pollinator network starting to be created. Meanwhile, some of the other work that we are beginning and starting to work on relates to what we found out when we were working with honeybees in the UK. Because what we saw with honeybees in the UK is through the seasons, you can have these periods of resource limitation. So we were interested in seeing, does that also happen somewhere like Greenland? So what pollinators are using through the season, but also where are there points where there are pinch points where networks could be more vulnerable? And so in order to start to answer that, we've got a data set which Thomas Roslin had collected on phenology of plants in great detail, just at one site in the south of Greenland in Nuuk. So we have phenology data from 2021 to 2024. We then, with my research group, collected pollinators each month in 23 and 24 at these same sites. So once this data is analysed, we can start to look at how pollinator networks change through the months and over years, and also relate that to the phenology of the plants in much more detail as well. So again, that's a piece of work for the future. We also, again, Thomas Roslin's group had malaise traps at these sites. So we've also got a record of all of the flying insects, which you can identify with metabarcoding. And then the final kind of study area that we're just starting to look at, which I want to pursue further over the next years, is again taking an historical approach, a bit like we did with the honeybees again in the UK. But this time using the herbarium and the museum samples that we have. So our herbarium samples go back to 1739, but they are more regularly collected from the late 1800s to sort of 1900s is where we have a more consistent set of records. And they go through, not to the present day because of a lot less botanists collecting, but they go through pretty reliably till around 2010 or so. And so one of the things that we're starting to look at is some of the key flowering plants for pollinators, and has the phenology of their flowering changed over historical time. So we have some Erasmus intern students at the moment who have spent many hours coding plants from the digitized specimens to see whether they were in flower or not, and relate that to the year the plants were collected. We can also potentially do genetic studies going way back in time with these herbarium samples. We have good samples of bees and flies, which also go back in time. And we can also potentially take pollen from the bodies of those and metabarcode that to see what plants were being used. These are pictures of bumblebees here, which is a bit misleading because there's only 2 species of bumblebee in Greenland, Bombus polaris and its parasite, and you don't see very many of them. So mostly this is tiny flies, which we're actually sampling in Greenland. But we can start to look at, has phenology changed, and is there a mismatch between the availability of plants and pollinators? And then just to finish up, and we just did a bit of a kind of test really, just using one set of herbarium records. And this is the oldest herbarium records that we have from 1739. And this guy, Paul Ligel, was a missionary. His father was a missionary before him. And he was based in Kisiengerit, which is one of our study sites. So we took a record of all of the plants that he collected in 1739, and we went back to the site, and we knew that he had collected his plants as part of his missionary work. So he wasn't going far away. He was walking in the settlement of Kisiengerit and then going to where the chapel was. So it's a small local area. So we actually did a kind of mini survey within the same area. This is the site here. And we looked at what plants we could find, and could we re-find the plants that were within Paul's herbarium. And actually we could re-find nearly all of them. They were all common plants that you find growing around houses and around the villages and in the tundra, which was immediately surrounding the community. And just about all the plants we could then recollect. And this was just as a kind of public engagement exercise, but it also means the potential to look at how the genetics of plants and how those have changed over time is also potentially possible if we can recollect at these quite precise sites. Okay, so that's it from me. So thank you ever so much for listening.
Thank you, Natasha. Are there any questions?
First of all, fantastic talk, really interesting what you have been doing and currently are doing. I myself am really interested in pollen and studying pollen and most of these type of stuff. But I've recently kind of read into the literature with biology and pollen, and also realized that as you always report amounts of breeds been posted that there's a problem with quantitative assessment of pollen using metabolic coding. So you kind of can get a understanding of how much there might be, but it's difficult to get a certain percentage of how much plant there is in samples, for example. But how have you addressed that in your years of research? Because you've for many years with putting down.
Yeah. Yeah, so there's been lots and lots of research done on this. And so it's kind of got to a consensus now. So it kind of went from, so lots of people were doing kind of validation studies with metabarcoding and lots of different systems. And those studies ranged from, it's completely unquantitative to there's a complete correlation, like, so very, very variable. But now there's been so many studies using pollen metabarcoding. This is different to, say, eDNA, which is a whole different bag of fish, okay? But for pollen metabarcoding, there's often a pretty good correlation between, so for example, some of the studies were looking at pollen collected in the air, where they had done microscopy studies and then a DNA study next to it. And there's a pretty good correlation between abundance. But there's lots of biases all the way through the DNA extraction process. So it means that it is not a, you can't use the read count as a quantitative measure per se. So all of those results that you saw there are all relative abundance. So you basically just take a percentage. So you can't necessarily say, okay, this amount of DNA relates to this many pollen grains, but what you can say is when you look across all of the sample, there is more of this than there is of this. Some of the studies, as it's kind of gone through its kind of journey with this, then stated, well, if it's not totally quantitative, maybe you should just use presence absence. But actually, that's more misleading. So if you think about something like, if I go back to that one, yeah. If you use presence absence with this data, it's much more misleading because what most pollinators do is they have major plants and minor plants. And if you use presence absence, then you overinflate the importance of the minor plants. So something like with the honeybees here, within this little black line here, this is taxa which are used below 10%. And there's actually quite a lot of taxa in this. And that's going to be a mixture of real things that the honeybees are using, but potentially also things that they've kind of flown through a field and got pollen, you know, wind pollinated pollen on them. So the things they're using in small amounts we're less interested in. So it's important not to use presence absence and then inflate those. So we did a big review paper where we got all the people doing pollen metabarcoding, all the different studies that they had done. And our consensus was that you should use relative abundance as your measure. And then you have an idea of how quantitative, it's more of this compared to this, but you're not saying it's something which is a direct quantitative measure.
Did you know, I was really interested in the first part of the talk when you talk about these barcoding efforts for entire floras.
Mm-hmm.
And this, the UK being like one of the first countries to do this at a national level. How many other countries have followed with this effort, like with similar projects? Are there now many countries where the entire flora is barcoded?
Yeah, lots. Yeah, and it's really valuable because when it came to Greenland, we want to make a broader like genome reference library, but that's going to take years. Meanwhile, we've got, you know, pollinators already. We want to make a start. And so what we actually did once we had our species list, the whole point about barcodes is they should be consistent for the species. So if that species has been collected somewhere else, it shouldn't matter. And because we had done the UK, there has been a big effort in Norway with Norbol. Canada has done a huge number at Guelph of their Arctic plants. Our colleagues, again in Tromsø in Norway, had also done samples. So once we then had our species list, we drew down all of the barcodes which were already available on online sources, and we could actually do the whole of Greenland. So now we're getting such good reference libraries of whole countries. It then enables that as a global resource to be used. And there's a big initiative now continuing. So it's called IBOL Europe, which is a big EU-funded project to continue DNA barcoding, and to run alongside that, also having wider sort of genome representation of plants as well. So that's kind of continuing. So now a good number of countries have got like pretty good barcode libraries. The tropics, as you can imagine, are still just such high diversity. And the key thing about a barcode library, and the reason why it was easier to do it in Wales, for example, is because you build on generations of botanists, because we use mostly herbarium specimens. And we use those specimens from collectors that we knew their plants. And you can go back easily 50, 60 years with a herbarium specimen and treat it as a fresh sample. So 80% of the specimens for Wales came from the herbarium. And so you basically have the collected effort of generations of botanists and their work identifying the specimens in order to make a reference library. So it was kind of a historically collaborative project as well as one sort of in the present sense. Yeah.
I can just follow with something that you said that was also in your talk, was how important these herbarium and museum specimens have been and are for understanding things like the response of organisms to climate change. But you also pointed out that now there are fewer people collecting herbarium specimens. So is this something that we should worry about, that we're sort of so interested in other forms of doing science, and this preservation of specimens that can be used in the future for other applications is not being done at the moment?
Yeah, because it's so important. So many of the traditional uses of herbarium specimens were for taxonomic research. And obviously you need enough samples to do your taxonomic work. But all of the use of these specimens for global change obviously relies on a timeline of specimens with good sample sizes of species. And basically you see within most herbarium, basically just a drop off often from 2000 onwards, where we just don't have that data available. So we have this amazing ability to go back in time, and then we kind of miss the kind of current time. We're quite lucky in Greenland that there was a long-term set of surveys that were done over many years, collecting the plants and animals of Greenland, but that then stopped in the late 1990s. Within Denmark, there was also the Atlas Flora Danica, which was done. And as part of that process, they collected herbarium vouchers. So as a comment, while the Atlas is being made here in Sweden, if you can collect vouchers, that's super valuable, because they were actually collected to help with the identification process. But it meant we have really good recent specimens because they were collected to check as part of the Atlas. And we also have amazing collections of the microspecies groups. So amazing rubus and Traxacum collections and things like that, which are again for identification purposes, but we can now use those for global change research as well.
Okay, you mentioned that we have a little bit of different history in Sweden because our gap is in the 60s and the 70s. And now the collection are at that level. So the museum has problem to take care of the material.
Yes.
Because so many of the NGO botanists especially collect so lots of specimens. And also lots of diapomictic groups, Eurasian, Taraxacum and so on. So it's so for the museum's part, it's more a problem of too much, not too little. Because they have not people to take care of everything.
Yeah, And it's really interesting. So the previous curator to me wanted to throw away all of the ecological samples, as he calls them, or the microspecies, because his view was that the herbarium should just be a representation of the, you know, the overall diversity. But then I come in as curator, who is a global change researcher, in terms of my use of the herbarium. Suddenly I'm like, no, all of it. You need to keep all of it. What we do, we compromise. So for the Atlas Floridanica material, a lot of it hasn't been mounted yet. And so we will mount some of it and we will keep quite a lot of it unmounted. So it's got its identifications on, because we don't have enough resource to mount all of the material. But we will keep all of it so that we can do these types of studies. Because also the resource, like Atlas Floridanica was done over a kind of 20-year period, and the amount of labor and resource that went into that, and obviously we have the distribution records as key output of that, but we do also have these collections which are incredibly valuable. But we do also draw the line. So I mean, we have an awful lot of inquiries every day of people, say, wanting to donate various herbaria that they have, or school herbaria. We've inherited all of the individual herbaria of all the university departments, you know, within Denmark, like, you know, we inherit all of those. But we do, which is nice, have really good support of that. So there is a good understanding of the importance of that collection. And we're building or planning, starting to plan new collection facilities in the future, which will enable us to keep these. We also, it's 2.3 million specimens in Herbarium C. So from our point of view, like a few more, like, you know, it's such a vast collection anyway, which we're trying to digitize all of that at the moment. And we've digitized Greenland and we're digitizing Denmark at the moment. But it is on quite a big scale that we need to work in terms of our facilities for the future already.
Super interesting all around, but I thought that this thing about the June gap was really interesting, and I was thinking about, like, you mentioned it's kind of a period of resource limitation, and I guess probably just stress with the beekeepers. So first of all, like, why do they say it's a resource limitation? Because the bees are, like, less active, or what is the? I guess what I get at is that just because they're going to more diverse flowers, I mean, we don't, if you're just looking at what's the pollen on the animals, then we don't know what's available in the landscape. So maybe they're just choosing different things. And so do you have other information about, like, what's actually available in the landscape at the same time? And are they limited, or are they just diversifying?
Yeah, and that's a good question, because I've kind of skipped quite a bit of the kind of backstory for kind of, you know, brevity. So the two other sources of information that we have for these conclusions is we also measured stores and the size of the colonies as well. So we measured the number of frames of bees to use a measure of colony strength. And then we also measured the level of stores, which is basically the number of frames of honey that the bees have available, pollen as well, but mostly honey. So we measure that over the months, and we see that this period where they're doing all of this with the plants is a period where their stores are limited. So the bee colony increases rapidly during the April and May phase. And then the colony still stays strong, but they use all of their honey resources, because which means that there is a resource limitation happening. So the resource limitation is measured through the amount of stores that they have available. We then look, oh, well, okay, what's available within the landscape at that point? And not in the actual paper, but what we were also measuring at the same time, which was a kind of crazy thing to do, because this is a huge area. So, like, this is in acres. It's like 500 acres, like this site. But we got volunteers, and we divided up the whole of the botanic garden, the whole of the nature reserve into zones. And each month we got them to record everything in flower across the whole of the botanic garden and nature reserve. So we had an idea of what was in flower. Not necessarily resource availability, because we didn't do measures of, say, nectar levels or numbers of flowers, because it's such a big site, but we've got a pretty good idea of area of something which is in flower over that time. And that, along with the measure of stores within the honey, was that, okay, there's a period of resource limitation here. We know that these plants, which they were using here, aren't flowering anymore. And so this is the pattern that we see. So from that, we then concluded that, whereas, sorry for June. So from this, at first, if you just see this pattern, we were thinking, no, they're just doing loads of things because more things are in flower. So maybe they're using more things. But then when we looked at the level of stores and what was flowering within the landscape, we concluded that it was a resource limitation period from there. Yeah.
Any other questions?
So is there a very good correlation between what they use and what is available, or is there clear preferences? Say, for instance, this Rubus fruticosus, is it just availability? Why it's so popular, or is there something specific about that?
So no, there's no correlation. There's no correlation between overall availability in the landscape and what they use. So you can kind of see pattern, and it seems to be a pattern made up of, they do use things which are abundant. So it is abundant things that they're using, and it's also things that have a long flowering period that they're using. But it's not just dictated by like literally the area covered. It's a little bit difficult to measure, though, for the reasons that we just said, that we measured just the overall area which was in flower rather than actual availability. And the other challenge that we have with our DNA barcoding is we are identifying pollen, but we don't know whether that pollen has come from nectar foraging or pollen foraging. So you're kind of getting a bit of a combination of both of those things in the results. We think a lot of this is probably driven by pollen, particularly early in the season, and then it's probably nectar resource later on. So we're also seeing a bit of a combination of factors. So in some ways, if we don't see a correlation between just broad availability, it could be for other reasons. It could be that we're not dividing into pollen and nectar, and we're not measuring exact amounts of nectar or pollen and things like this. But some of our colleagues that we've worked with at Bristol University have been doing that in more detail, and they worked with bumblebees in farmland, and they measured nectar availability as well, and then did meta-barcoding of bumblebees with us. And they also see a gap in June, which doesn't relate purely to availability in their case either. So our data kind of gives a bit of an indication, and kind of theirs sort of also supports it along the way.
Any other questions?
Yeah, thank you for the presentation. It's about the project in the Arctic.
Mm-hmm.
So as you say, Arctic environments are evolving faster than. So these increasing temperatures are causing that there's no melting faster as well. So the main consequence is that there are longer springs now. So some studies have found that plants now are flowering earlier than in the past. I was wondering about this mismatch between plants and pollinators, because one of the response of the plants is that they are advancing their flowering time And then this means that also pollinators should track all these plants, but also they could mismatch the main period of flowering. So, and yeah, I was just wondering, what are your expectations about these network structures changing across time?
Mm. So there's been a really interesting study, which we're kind of using as our hypothesis for this. There's been a few studies. I probably can't find it quickly enough but there's like a really nice set of data which has looked at it's not over historical time, it's over the last kind of 20 years from permanent plot data. So these kind of long-term experiment data sets. And what they've shown in Greenland and also the Canadian Arctic as well is that the most important factors for plants early in the season is snowmelt. And so snowmelt is the key factor at that point. Later in the season, it's temperature. It's like the overall summer temperature, which is really important. And those two things, although they kind of relate to each other, they don't completely relate to each other. So sometimes snowmelt can actually be a bit, and the plant's response is also dependent on when they flower in the season. So for early season plants, they will only start getting going when the snow melts, and they're not that temperature sensitive. So they're not that fussed when the temperature is whatever it is, as long as the snow melts, they go ahead kind of thing. The late season plants are really sensitive to temperature. So they flower earlier with temperature. And so what these studies have shown is that the early season plants flower whenever the snow has melted, and that's not necessarily kind of earlier, whereas the late season plants flower earlier because the temperature is higher. And you get this like shortening of the flowering season on a community level. So you actually see that the period where you've got lots of things available for pollinators has got narrower. And they've shown this now in quite a few different studies. So our hypothesis with the historical work is to look at, does that also take place kind of further back through historical time? So do we see a kind of an increase in flowering season over time? More recent data, though, is kind of coming out with more of the conclusion that you don't just see an increase in temperature, you're just seeing these incredibly like just up and down stochastic events in the Arctic. And so it could be that there is no simple, pattern to be discerned. It's just like almost chaotic in terms of what's happening. So they're the kind of hypotheses that we're kind of balancing against, that you might have a shortening of the season, in which case would the pollinators all kind of squeeze into that season as well? Maybe they can, they're very adaptable. But also if the season is just quite chaotic, can pollinators deal with that? And again, possibly they can, because they are used to these extreme conditions. And that's some of the kind of questions that we want to look at with the Arctic work.
I think we should stop here, but let's thank Natasha once again for a great talk.